The overall goal of this procedure is to initiate new neurites, rapidly extend them, and precisely connect them to target cells. This procedure is only possible by using cells grown in a controlled and reproducible environment.
This method allows us to answer key questions in neuroscience. What limits the growth rate of neurons? How fast can they grow? Do mechanical cues play a role? We can also study how synapses are formed. How do neurons find a target cell? How do synapses age, mature? And thirdly, we can start building systematically neural networks, study how neurons compute.
The main advantages of this technique are that it enables easy micromanipulation, experimental reproducibility, and ultimately a high degree of control at the single cell level.
To begin this procedure, clean and prepare the desired number of sterile cover slips. Next, coat each cover slip with 0.5 to 1 milliliters of 100 microgram per milliliter PDL for at least 2 hours at room temperature. After that, wash the cover slips twice with water and remove all the liquid. Let them dry in a sterile environment for 5 to 10 minutes or until the surface is completely dry. Then, place the microfluidic devices with the patterns facing up under the UV light in a biosafety cabinet for 10 minutes. After 10 minutes, place a microfluidic device with the pattern facing down in contact with a clean cover slip. Use the tweezers.